Hello, Ms. Coleman. My name is Tanvir Rashid. I'm doing this bonus assignment loan. It's hard to get people over for Thanksgiving, but whatever, I'll just do it. So basically, I'm choosing the DNA topic. So starting with history of DNA. Well, it started back in 1928 and then 1943, but I think the important stuff starts from 1950 when Edwin Chargaff, um, he was a biochemist. He studied the chemical composition of the DNA molecule. Uh, Chemical studies of DNA revealed it had a type of sugar called deoxyribose. Um, Deoxyribose, uh, that's for DNA, plus a phosphate group and four different molecules or bases called adenine, thymine, guanine, cytosine. Uracil is an RNA that substitutes. um, Charger realized that the amount of adenine in DNA sample always equaled the amount of thymine. There was a equal ratio there, and the amount of cytosine always equaled the amount of guanine. So that's the reason they're always related. So this was called the Chargaff's law. It helped scientists study the structure of DNA. And then moving on to 1952, this will sound familiar. Rosalind Franklin, yeah, her. She used a technique uh, with X-rays that focused beams of X-rays to produce photographs of the crystallized forms of molecules. Uh, she was able to find the 3D structure of the molecule. So Franklin's image of DNA ultimately revealed its exact structure. So by viewing the X-rays, she could see what DNA structure looked like. And this was very popular and very important back then because it was still evolving DNA. So this is an important discovery. And then 53, James Watson and Francis Crick stole her idea, stole Franklin's Rosalind idea. Um, so they viewed Franklin's X-rays diffraction image without their knowledge. They figured out that DNA is made up of two mirror strand composed of stacked DNA nucleotides, a sugar and a phosphate group, plus a base that resemble into double helix, twisting double helix. So the double helix, it's formed from stacked nucleotides, it resembles a ladder. So so basically, yeah, they stole Franklin's idea and they got all the credit. They won a Nobel Peace Prize. Unfortunately, Franklin died in '58, couldn't receive it. Um, anyway, then the next up in '75, Frederick Sanger, he uh, he developed the chain termination method of DNA sequencing. So basically, different size fragments of DNA are produced and separated based on their lengths. So scientists can figure out the exact sequences of A's, T's, adenine, thymine, cytosine, and guanines in a molecule. And this is known as Sanger. Um, then in 77 and 79, 1977 and 79, scientists at Genentech, the world's first genetic engineering company, were the first to genetically modify organisms to produce human hormones, somatostatin, human insulin, and human growth hormones. So molecular biologists have since used genetic tricks to produce many drugs that benefit human health. So this stuff benefited human health. But then basically that covers the basics of genetic the history of DNA. And let's move on. So the functions and structure of DNA. Well, the structure of DNA. DNA is composed of polynucleotides. Um, it has a phosphate group, deoxyribose sugar, and a nitrogen-containing base. Bases, basically. There's four of them: adenine, thymine, guanine, cytosine, or cytosine and guanine, whatever. Um, so it also has a double helix that is formed by hydrogen bonds between the polynucleotides. That makes it a double helix and twisty ladder-shaped, uh, you know. and is densely fitted, compacted into chromosomes to fit into nucleus. Um, so DNA has purines, which are base that has double ring structure, adenine and guanine. Uh, adenine and guanine. So those are purines, and pyrimidines are thymine and cytosine. Well, what are the functions of DNA? Well, it stores an organism's genetic material in the nuclei. In the nuclei, it replicates itself when dividing. It provides code for template for the particular sequencing of amino acids that bond together and make a protein. It does everything for your life. DNA is life. That's as simple as that. Well, DNA and forensic science. That's really interesting, right? Well, it all starts with um, anything: blood, semen, saliva, urine, feces, hair, teeth, bone, tissue, and cells that's left over in a crime scene to determine, and these are all used to determine the DNA profile. Um, Anything contains DNA material inside a crime scene, starting from tissues to anything, to your cell phone, anything like that. So basically, whenever something goes wrong, the FBI comes over and they have their lab workers, a crime scene investigator during then, and they collect the blood samples that left over, or any sample, DNA sample, hair sample, even devices or any matter that's left over. They bring it to their lab and they test it for DNA, and if there's anything left in there, they take measures of how much is present in the crime scene, and they go on from there. Um, there is a different way. There's many ways to test it. The low-level DNA is referred to as touch DNA. It's collected from a victim's skin, um, where they were handled roughly. This may be helpful when examining evidence when it comes to difficult to retrieve fingerprints, such as gun handles or automobile dashboards. So yeah, there's also this stuff is really scary when but scary and also really cool. Um, who conducts DNA analysis? DNA working behalf of the FBI, National DNA Index System, um, any accredited organizations that the FBI hires. They must have good reputation because they do this for a living. Um, well, DNA testing is performed in a laboratory with highly dedicated equipment. They're talking about life or death and danger to this world, stuff like that. So they need to be very efficient and really smooth with how they are. So they have really good material on there. Well, who's tested in a forensics incident or crime scene? Everybody's tested. They want to see who did it, where were that person, who's all involved. So they'll try to get everybody DNA testing. Anyway, DNA forensic science is really cool and really interesting because it's really cool how a piece of hair can lead to you or lead to anybody, even me, even anybody. So um yeah, that's about it, Miss Coleman. Um, Getting ready for this last stretch of semester. Hope you have a good time. Bye.